subunits. And essentially, you can see that the ACC family is expressed fairly ubiquitously throughout the nervous system, both in sensory neurons, in motor neurons, in interneurons, and in muscle. So this really suggests a widespread role for acetylcholine as an inhibitory neurotransmitter in CLS. A quick distinction I'd like to make is that LGC48 is a little bit different than the rest of the ACC subunits in that it is only expressed in a pair of neurons in the tail. So most of these other subunits are expressed in a large percentage of neurons. And LGC48 is only expressed in a pair of neurons in the tail, and that will become important a little bit later. So now I wanted to focus a little bit on the roles that ACCs have in behavior. So I specifically looked at ACC1 in the pharyngeal motor circuit because I found that ACC1 is expressed in pharyngeal motor neurons, and we know that ACC1 is capable of forming a homomeric channel. So this is just a simplified pharyngeal motor circuit. The pharynx is the eating organ of the worm and is a muscle. So when the pharynx contracts and relaxes, it allows the worms to ingest the bacteria that is their food source. This muscle contraction and relaxation can be altered by MC, which is the excitatory motor neuron in the pharyngeal motor circuit, and M3, which is the inhibitory motor neuron in the circuit. And specifically, I was interested in the role of M3 in the pharyngeal motor circuit, and specifically M3 will cause the muscle to relax. And so I thought that if you removed ACC1 from M3, we might get a difference in the timing of the pharyngeal muscle contraction. And the way I studied this is by using an electropharyngogram. So electropharyngograms are extracellular recordings of the electrical activity in the pharynx. Basically, you take a microelectrode and you suck the very tip of the worm's nose into the electrode, and you basically measure the capacitative current that's flowing in and out of the mouth of the worm in response to changes in the pharyngeal muscle membrane potential. This is what an EPG trace looks like. So when the muscle membrane potential, so basically every time the muscle membrane potential changes, you'll get a spike. You get an upward spike when the muscle depolarizes. These are the inputs from that inhibitory motor neuron M3. They're called inhibitory postsynaptic potentials. And then when the muscle repolarizes, you get a downward spike. And basically, I was interested in the timing, so how long it took from peak to trough for the muscle to contract and then relax. And I thought that that would be a readout of how strong M3 is functioning. Because if M3 is functioning or is firing a lot, you get lots of IPSPs, and that would shorten the duration of the muscle contraction. And this is in fact the case. So when you remove ACC1, you have shortened pharyngeal muscle contraction due to loss of inhibition on the inhibitory motor neuron. It's quantified here. So you have wild type levels, and they're decreased in the ACC1 mutant. And this can be rescued. So when you reintroduce the wild type copy of ACC1, the pumps are now lengthened, and they're not significantly different from the wild type length. Moreover, when you introduce the rescue construct into wild type, you can lengthen the wild type pumps beyond normal wild type lengths, suggesting that overexpressing ACC1 can over-inhibit the inhibitory motor neuron, which results in lengthening pharyngeal pumps. So just a quick summary, you have cholinergic inputs onto ACC1 that's expressed on MC, I didn't talk about that, but then on M3. And when you remove ACC1, these neurons are allowed to be more active, resulting in potentially more pumps, although that's not something I measured, and shorter pumps. Okay, so the last part of my talk is investigating whether the ACCs are good targets for antiparasitic agonist drugs. So there are two main ways, in my mind, that you can affect channel function. The first way is to inactivate the channel. And so what happens here is that if you picture that synapse, what you're doing is preventing the presynaptic cell from communicating with the postsynaptic cell. So postsynaptic cell can still function otherwise, it just can no longer receive communication from that presynaptic cell. On the other hand, if you overactivate the channel, you're opening the channel even when its endogenous ligand is absent, and this not only results in abolishing communication between the pre and postsynaptic cell, it actually renders the postsynaptic cell non-functional. It is no longer able to function as a signal transducing neuron, and so this could have very deleterious effects if you affect a large proportion of the nervous system. And actually, in fact, one of the most successful antiparasitic drugs uses this strategy. So ivermectin is, like I said, a very, very successful antiparasitic drug, and it targets glutamate-gated chloride channels. So this is a picture of a glutamatergic synapse. Under normal conditions, glutamate is released from the presynaptic cell, binds to these glutamate-gated chloride channels, and transiently causes them to open, resulting in changes in electrical activity in the postsynaptic cell. Ivermectin binds to these glutamate-gated chloride channels and causes them to be permanently open. So these channels will be open regardless of whether or not their ligand is present, and will remain open once ivermectin is bound, resulting in constant chloride influx into the postsynaptic cell and essentially rendering that postsynaptic cell non-functional. Because the gluCLs are expressed in essential neural tissues, when you inhibit all the gluCL expressing tissues, it is lethal to the worm. And so what I wanted to do was exploit the similarities between the ACCs and the gluCLs. So they're both chloride channels. They both allow chloride through their core and result in the same effect on the postsynaptic cell. They differ in their ligand, but they don't differ in their ion selectivity. And so that's what I wanted to take advantage of. What I decided to do was to manipulate the ivermectin system. So this is just a picture of a wild type worm. I have the gluCL endogenous expression in green. And as I said, the subset of gluCL expressing neurons in the worm are essential. And so when you put these worms on ivermectin, that essential subset of neural tissues is inhibited and the worms die. We have a strain, JD369, that has, it's a glutamate-gated chloride channel mutant. It lacks all 4 endogenous ivermectin targets. And so these worms are completely insensitive to ivermectin. They are lacking the functional target for the drug and are very resistant. They're viable, they're fertile. They actually only have very subtle phenotypes. And so what I wanted to do is selectively reintroduce the gluCLs into the ACC expressing tissues. So now, instead of having gluCLs expressed in their endogenous tissues, I would only have them expressed on ACC expressing tissues. And this would result in a subset of neurons or neural tissues in the worm that now have an activatable chloride channel on them. And when I expose those worms to ivermectin, that's here, I can essentially mimic a drug that directly overactivated the ACCs by activating a chloride channel in those same tissues. As a control, I used the LGC48 promoter, which I mentioned was one of the ACC subunits that only had expression in non-essential neurons. It had just a pair of sensory neurons in the head and the tail. So we thought this would be a good control because these worms should be able to survive very well in our And this is in fact the case. So these are pictures of plates that I put eggs on. They're 50 nanograms per mil ivermectin, and I put eggs on them and then waited 3 days to let them let the worms develop to adulthood. And you can see here that while the control strain is developed very nicely, the ACC1 strain actually develops, you know, they do develop, definitely not to the same extent, but they escape the first larval stage and continue their larval development. The other strains really have basically no survival. And I quantified this as larval escape. So this is the first larval stage. So when the eggs hatch, they hatch as L1s and then normally go through a subsequent larval development. And I was just scoring for any worms that were older than the first larval stage after 3 days. And the control strain does very well on up to 15 nanograms ivermectin, and all the other strains do quite poorly with ACC1, as I mentioned a little bit ago. But this suggests that the ACCs would be good drug targets